How complex exactly is a tRNA molecule, and how could you possibly be able to organize and store all of its associated sequences? Transport RNA, or tRNA, acts like a cloverleaf-shaped mailman. As messenger RNA is read by the ribosome, tRNA does its deliveries, attaching its anticodon to a matching codon on the mRNA, binding or dropping off its specific amino acid in the correct order to produce a specific protein. The specificity of amino acid attachment to the tRNA 3' binding site is determined by the amino acyl tRNA synthetase, a highly specific enzyme which recognizes and activates a specific amino acid and transfers it to the tRNA of the 3' CCA terminus. This terminus consists of cytosine, and adenine, and allows amino acids to conveniently attach via the amino acid's carboxyl group, charging or amino acylating it, so it's ready to participate in protein synthesis. tRNA have a unique complexity, as not only do they have the most amount of nucleoside modifications found in nature, but they also allow us to see how entire groups of tRNAs evolve together over time, as different organelles and cells possess varieties of tRNA molecules that are similar to each other but still have unique characteristics. So how would we construct a tRNA database that is user-friendly and addresses all of tRNA's structural and functional nuances? This is Frank, the geneticist, who is accessing tRNA databases for some important research he is conducting into mitochondrial genetics. His first search brings him to the Mammoth tRNA database, a detailed mitochondrial tRNA database, allowing Frank to analyze specific anticodons present in his mitochondrial tRNA sequences of interest. Frank was also drawn by its user-friendly design and engaging color codes and visualization style. Frank's next search brings him to the BLAST database. This database allowed Frank to enter his tRNA sequence of interest and find similar or homologous sequences in return. Using these sequence similarity comparisons, he can identify tRNA genes and their interspecies variants, helping him understand tRNA gene variations between different organisms. But what if Frank wasn't interested in mitochondrial tRNA, and wanted to look into cytoplasmic or chloroplasmic tRNA instead? Well, that is where the gap in research lied. There was yet to be a relational database that compiled all tRNA genes and sequences into state-of-the-art IT infrastructure, which was highly flexible and user-friendly. So, in 2009, Frank Juling and his team set out to design a tRNA database that would combine all the best aspects of existing genomic databases to form the ultimate and complete tRNA database. They began their complex journey with Excel spreadsheets, which stored the original tRNA sequence collection. But having a bunch of numbers and sequences in Excel documents wasn't user-friendly, so they retrieved the complete dataset and stored it in index tables. These index tables function with the user first inputting a query, which was sorted into an index that was then matched into associated columns. After verifying the integrity of each sequence within these tables, they transferred all this data into a relational database system, which is essentially just a digital filing cabinet which can link and connect data tables and find data easily. But this was just one piece of the puzzle, as users will have to have any taxonomic queries answered accurately and rapidly. As such, the researchers implemented a taxonomic tree from the National Center of Biotechnology Information, or as most of you know it as, the site you go to when you want to find quality studies. But what if during his search for tRNA sequences from, say, the Homo genus on the new tRNA database, Frank entered the taxonomic ID 9606 instead? Well, the researchers took this into consideration, and they allowed for synonyms, taxonomic IDs, and strains to all be recognized by the query system. The query system used the advanced functionality provided by MySQL, which is a relational database management for web databases. By using MySQL open source program, the researchers were able to combine it with the BLAST and Mammoth tRNA-based database frameworks to build a query system that provided results for queries regarding DNA or RNA sequences, anticodons, amino acid families, gene descriptions, and structural characteristics. To polish off any inconsistencies in the dataset, a new color code was assigned to the CCA ends, since the Mammoth tRNA database gave them data that didn't include CCA triplets, as mitochondrial DNA doesn't encode them. And to ensure user convenience, tRNA secondary structures could be viewed via a thumbnail. Additionally, its domains and nucleotide positions were presented using an image generator. Finally, any retrieved data could be downloaded and exported in a variety of file formats, allowing him to continue developing his research in other applications. So, after all this, what did this new tRNA database entail for the field of genetics? Well, it provided researchers with a centralized dataset, making comparative studies more accessible and efficient. The functional annotations that are provided in the database, such as unique amino acids carried by certain tRNA anticodons, also allow for analysis of modifications and translational roles that certain tRNAs had. However, the new tRNA database isn't perfect, and future researchers can work to fill in the gaps. such as including the cataloging of pathological tRNA mutations, which can enhance the identification of disease-causing mutations, helping predict disease risk. But for geneticists around the world, the 2009 tRNA database works perfectly for exciting research into genetics, and one day it might serve you well too.